I am really delighted to be introducing the 2023 Edwin G. Conklin Medal Award winner. Conklin himself really embodied the qualities we see in the medal awardee, being both a remarkable developmental biologist and a wonderful mentor. So we've been giving this award since 1995. I'll say just a little bit about Conklin. He really spent most of his research career at the Marine Biological Lab, like many people here have been influenced by, and his research focused on lineage tracing in the Ascidian Stiella, which has this incredible yellow crescent that can be used as a natural lineage tracer. And that allowed him to really get into experimental embryology, which was something that was considered quite risque at the time. So Conklin went on to be chair of the biology department at Princeton for 25 years, where he built a strong department and mentored countless undergraduates. And throughout his career, he remained fascinated by the events of early embryogenesis. So it's very fitting that that is also true of this year's winner of the Conklin Medal, Lilianna Solnica-Krezel, from Washington University School of Medicine. So Lila grew up in southern Poland during the communist regime and moved to Warsaw at 18 to study biology and went on to get her master's degree there. We see her here doing what she tells me was probably one of her very first experiments, helping her brother with some experimental work. She and her brother were both first-generation students, by the way. After she got her master's, she took the long route from Warsaw to Wisconsin, getting her PhD at the University of Madison, Wisconsin, where she studied the slime mold, Physarum polycephalum, with William Dove. While there, she was a genetics TA for Bill Dove and learnt about the zebrafish, which she became fascinated with. And that led her to go on to be the first postdoc in Wolfgang Drever's lab at Mass General Hospital in Boston. She's the stripy shirt on the right, and I do remember that at that time we all wore lots of stripy shirts. Now, in Wolfgang's lab, she was one of the drivers of the large-scale genetic screen in the zebrafish model that was going on there. And as many of you know, there were two parallel screens running, one in Tübingen in Germany and one in Boston. And together, they led to a remarkable assortment of mutants that were described in this very special issue of Development published in 1996. Lila's an author on all 11 of the papers out of the Boston group, and this is one of her first author papers describing gastrulation mutants. That same year, 96, she went to Vanderbilt as an assistant professor, and she, in her career, used those gastrulation mutants to really give her an inroad into studying the cellular behaviors and morphogenetic processes of gastrulation, also isolating genes that were involved in the process, the mutants, in fact, that was very non-trivial at the time. And through this, she became one of the founders of the field of vertebrate planar cell polarity signaling. Her work continues to uncover new details about how PCP signaling promotes polarized cell behaviors, and she's also had major contributions to related areas such as axis development, organogenesis, and some forays into disease models too. Now at Vanderbilt, she became full professor in 2005. In 2010, moved to Washington University Medical School as chair of the Department of Developmental Biology, and along the way has earned many honors, including very recently election to the American Academy of Arts and Sciences. She's also been a past president of both the Society for Developmental Biology and the International Zebrafish Society. Now, I should say that her nominators talk not just about the research, but also a great deal about her mentorship. Her nomination was supported by a large group of peers, junior faculty colleagues, and previous trainees. And they really emphasized her depth of engagement as a mentor and how she had brought out their best selves. I also learned that her trainees call her the zebrafish lady, and looking at the images available of her online, you can see why. So as of last year, Lila had launched 15 postdocs and 19 PhDs out of her lab. The support letters, like these lab photos, paint a picture of deep personal investment with the trainees, many of whom commented she is their personal role model. I asked one of her lab members if they had any fun anecdotes about Lila, and I'm going to share this one directly. I'm going to quote it because I think it tells you a lot about her, and don't be nervous, Lila. So, okay, quote, I have a friend who is very social, very smart. He loves gossip. He met up with his friend of similar proclivities at a meeting. It may have been the annual SDB meeting. They sat at the entrance to the dining hall and shared their treasures of gossip as each PI walked by. When Lila arrived, one turned to the other and said, "No gossip." Now that's actually a huge achievement. Now, I should say that as president of the society, I don't get to play a part in selecting the award winners, but it's a huge personal pleasure for me to be giving this award to Lila as a fellow zebrafish developmental biologist and someone who I've known throughout my own time in the field, which is now nearly 30 years. So Lila, please come up and let me give you your Conklin medal.
I feel that I was already awarded and won the moment I started to work on zebrafish, on gastrulation, and every day has been an adventure, and to be awarded for this is really remarkable. It really takes a village to move science forward, and I take this award on behalf of the entire zebrafish community. This is not to say that other zebrafish investigators should not be awarded in the future. And that's why I'm so happy that Kevin Thiessen is another awardee today. Oops. Throughout this meeting, we talked about various aspects of development. I think every aspect of development is fascinating. I would be happy to work on limb development, I would be happy to work on pancreas development, but we are all different. And somehow I was, and I continue to be fascinated by this amazing process of gastrulation, which, this is funny, which happens in all the animals, vertebrates and invertebrates, which start pretty similarly, but then during gastrulation look quite differently, yet during the pharyngula stage, they are very similar, suggesting that there must be conserved mechanisms. As Vicky mentioned, my road to gastrulation started in Poland, and I was a student just as the Solidarity movement was initiated. It was an amazing time in Poland. There was a lot of optimism. But the regime started to show its dark sides. Students went on strike at Warsaw University, and unfortunately, in 1981, in December, martial law was imposed. We lost hope that things will change in Poland, and this was part of motivation to just continue to focus on science. And as I was reading about science through current contents, for those older of you, remember, one paper arrived in Warsaw, and that was a paper in Nature by Janine Slein-Volhard and Eric Wischaus, who reported the results of a genetic screen for mutations that affect embryogenesis in Drosophila. And what you can see here, this is in Polish, because that was the first review that I wrote, scientific review as an undergraduate student, and I was completely hooked. Just on this concept that one can use genetics to dissect a very complex developmental process. And part of that curiosity motivated me to leave Poland. And as I was studying slime molds in Poland, I joined the lab of Bill Duff at University of Wisconsin. And Bill was one of these scientists who started with phages, then moved to slime molds, almost at the same time started to work on mouse and also on human genetics. And he was very encouraging, as I've been looking for something that I would like to study of my own. And of course, what I was really fascinated by was, unfortunately, the movies will not be playing, so that will be very sad. They are playing. This gastrulation is happening. Started to be fascinated by gastrulation and work of Ray Keller, but I really wanted to study gastrulation using genetics. So it was really either mouse or potentially zebrafish. And I was learning more about zebrafish. I was also learning about the work of Katrin Anderson. But when I went to Eugene to visit with Wolfgang Driver and met the Eugene group led by Chuck Kimmel, of course was familiar with one of the very first gastrulation mutants, such as spade-tail, that Robert Ho, who is here, has been studying. And when Marnie Halpern, who I hope also is here, showed me the first zebrafish embryos during gastrulation, basically there was no way back. It had to be zebrafish. So I joined the lab of Wolfgang Driver, and the first thing we wanted to do is to establish effective mutagenesis of the germline. And we compared EMS and ENU, along with Mary Mullins in Janie Nyslein-Volhard lab, showed that it is ENU that is a more effective mutagen in zebrafish. That led to quite a lot of effort by this group of people that Vicky already mentioned. What strikes me about this effort is that while we found thousands and thousands of mutations, it was actually a relatively small group of people in Janie Nyslein-Volhard lab and also in Boston. Wolfgang was an amazing mentor and also a very generous mentor. Those who screened, we were invited to take the mutants we were interested in. So indeed, one beautiful day, I packed quite a number of little bottles with little fish in them and boarded a plane. It was still possible at the time to do so, and landed at Vanderbilt. Vanderbilt was a great place for us, and I want to mention particularly three people. Chris Wright, who is here still at the conference, Bridget Hogan, a former president of the society. Without their support, I don't think I would be able to continue and succeed in science. And also Bruce Apple, my scientific brother, who became my colleague and with whom we're building fish facility in the zebrafish community at Vanderbilt. You see a picture of collection of gastrulation mutants, just a handful of them, a few good mutants, if you will. We didn't know what they were at the time. We didn't even have a genetic map of a zebrafish genome in these days, and that's why I say it takes a village. It was a concerted effort of the entire zebrafish community to build these genetic tools, to clone these mutants, to build methods to study their function in embryos. And I'll tell you some stories about what these mutants taught us about zebrafish and vertebrate development in general. This one turned out to be a transcription factor bosozoc. These are components of nodal signaling, as Alex Shear has shown, components of the BMP signaling, mostly studied in zebrafish by Mary Mullins, Matthias Hammerschmidt, and others. And we will talk a little bit about these non-canonical wind PCP mutants. The first story is about bosozoc, and it is about formation of the gastrula organizer. Of course, first described by Spemann and Madgold in frog. This is a zebrafish version from their extemple lab. When this embryonic shield is transplanted to the ventral side of another embryo, there is this process of twinning happening. So like in the frog, zebrafish organizer can induce an ectopic embryonic axis. And like in frog, the organizer is expressing guscoid, the gene that Ken Cho in Edith de Robertis' lab has cloned. If organizer is removed mechanically, or if beta-catenin is mutated in zebrafish, these phenotypes appear. It's a gradation of almost normal embryo, but also embryos that miss notochord, or miss head, miss trunk, or don't have axis whatsoever. Bozozoc mutant, as you can see, is actually similar to one of the milder versions of the beta-catenin phenotype. Toshio Hirano and this is extremely touchy-feely. Toshio Hirano and also Robert Ho has cloned this gene and shown it can induce embryonic axis. You can also see that it is expressed in the organizer. And we now know that it's a direct target of beta-catenin. When bozozoc is mutated, guscoid in the organizer is not expressed. So how does this transcription factor work to affect functions of the organizer? Beta-catenin is a maternal gene. It induces bozozoc. Bozozoc is the first gene to be induced during zygotic genome activation. And then bozozoc seems to be activating guscoid, but bozozoc is a transcriptional repressor. And via a very nice collaboration with Igor David's lab, we shown that bozozoc is promoting guscoid expression because it is first repressing expression of genes like VEGA, which are inhibiting guscoid expression. And similarly, bozozoc mutants lack notochord and also have smaller forebrain. Collaborating with Chris Wright, we have shown that this function of bozozoc is achieved by bozozoc inhibiting Wnt8, which is inhibit organ formation of the notochord, but also is posteriorizing forebrain. Another very important insight about how bozozoc works in the organizer came from a typical genetic experiment. In addition to bozozoc, gene cordino has been identified in Nisla Volhard lab, and Matthias Hammerschmidt has shown that it encodes cordin, inhibitor of BMP. When we made a double mutant, one of the strongest and most exciting double mutant phenotypes was observed. While cordin has all the parts of the body, just a smaller head, the notochord is present, and it has excess of blood and tail. The double mutants don't have a head, don't have a trunk, and don't really have a polarity. So from this, we concluded that bozozoc works largely in parallel to cordin. And also, bozozoc not only negatively regulates Wnt8, but also promotes expression of other components of the organizer, as we have shown with Chris Wright. So one gene, transcriptional repressor, is mediating quite a lot of functions of beta-catenin during organizer formation. Seeing how different was the shape of this double mutant was, it was very clear that BMP is not just about patterning, it must be about regulating cell movements. So a lot of effort in my lab and a lot of interest was on understanding how zebrafish gastrulation happens and how movements during gastrulation shape the embryonic body. And the beginning of gastrulation, as we knew from studies of Chuck Kimmel, we have a fate map of zebrafish embryo with endoderm, mesoderm, and ectoderm layered in the zebrafish animal cap. And during gastrulation, these germ layers are placed in the proper position, but what you can also appreciate, their dimensions change in a very dramatic way. While both endoderm and mesoderm are spread from the ventral to the dorsal embryonic axis, at the end of gastrulation, both endoderm and mesoderm congregate on the dorsal side of the embryo, and that's the movement of convergence and extension. So how these movements happen was one of the important questions in the lab. And these two short mutants, Knipek and Trilobite, gave us some answers. So the transgenic embryo here shows how these movements are happening. And you can see these convergence movements are bringing mesoderm, because these are mostly mesodermal cells, to the dorsal axis. We were very interested how these movements are patterned from ventral to dorsal. And I'll show you later that cells at the very ventral aspect of the embryo simply move to the vegetal pole. But when we look in the lateral region, these cells are not very highly polarized, and they move almost like amoebae towards the dorsal side. A little bit later during development, these cells become highly polarized and now move in a very organized manner towards dorsal. You can also notice that these are all mesenchymal cells that move as individuals. And in the dorsal region, we and others have observed the same type of mediolateral intercalations that pioneering studies from Ray Taylor have shown. Our mutants are short. They actually show all the... Okay. Okay. They have actually all the parts that embryos should have. Everything is just shorter along the anterior-posterior axis and broader in mediolateral direction, as you can see here in the somites and the neural plate. Looking at the notochord, you will notice that in the wild-type embryo, notochord cells are polarized, mostly in the mediolateral direction, perpendicular to the anterior-posterior axis. And in the notochord of these mutants, cells are extremely randomly polarized or not polarized at all. However, a little bit later during development, these cells become partially polarized, suggesting that in addition to activity of whatever these genes encode, there must be additional pathways that help convergence and extension. So as Vicky said, it was not easy to clone these mutants, but our group had a lot of very enthusiastic and talented people. And Jacek Tubczewski has shown that knypek, which in Polish means shorty, encodes glypican. Jason Jessen identified trilobite as zebrafish equivalent of Wangel, and that was a genetic evidence that this pathway is equivalent of the Drosophila PCP pathway. Already at the time, we had such evidence from John Wallingford, Richard Harland, and also Jim Smith and Steve Wilson's lab. Flo Marlowe cloned rho kinase and shown that it is also a component of this pathway. And more recently, using reverse genetics, we have demonstrated that adhesion GPCR is also a component of this pathway. So how does this pathway mediate cell movements during gastrulation without affecting cell phase? We have shown that cells still know where the dorsal is, so this pathway does not inform cells about the direction of migration, but it's important for cell polarization along the anterior-posterior axis. And Brian Siruna, when he was in Alex Shear's lab, was the first to show that prickle, which is a component of this pathway, is accumulating at the anterior sides of the cells. And we have shown that Wangel2, while present at all the cells, all the edges of the cell, accumulates at the anterior side of the cells as well, while disheveled is on the posterior side of the cell. Diane Sepic in the lab showed that these cells, which are mesenchymal, and that's also really contrast to all the other contexts in which PC pathway operates in epithelium, that this pathway is also responsible for polarizing microtubule organizing center, which becomes biased to the posterior side of the cells. Based on these studies, we propose that in vertebrates, PCP pathway serves as a compass, and as a compass that points in anterior-posterior direction. Of course, if this is the case, how does the compass know where the anterior and posterior is? Studies from Rudi Winkelbauer's lab, but also thesis collaborating with Chris Wright, suggested that activin or nodal pathway might be providing this information and anterior-posterior patterning. And very talented postdoc in the lab, Margot Williams decided to test this. And here we went back to the mutants and used crypto, or a one-eye pinhead, which Alex Shear showed is an essential cofactor for nodal signaling. And as had been shown, in the nodal mutants, or maternal zygotic one-eye pinhead mutants, the embryonic axis is extremely short. These embryos actually lack endoderm and most of the mesoderm, but neuroectoderm undergoes abnormal morphogenesis. And Margot shown that indeed cell polarity in these mutants is affected. And therefore we thought, okay, if cell polarity is affected, clearly nodal must be regulating PCP. And then Margot has done a killer experiment, which means that it's experiment that kills your hypothesis. And indeed, that's what happened, because Margot looked at localization of prickle, and in these nodal mutants, very, very unpolarized cells, prickle still recognize the anterior side of the cell. Based on these and other experiments, Margot suggested that actually nodal works parallel to PCP, suggesting that there must be other pathways that collaborate with nodal. And of course, whenever we run out of ideas, we do a genetic screen. So that's what we have done, still at Vanderbilt. Our former very talented student, Chunyu Yin, now at Cincinnati Children's Hospital, has done a synthetic screen in which we incorporated a weaker allele of Knipek, or glypican. And we identified so-called ugly duckling, which Hatsushi Sawada demonstrated by generating maternal zygotic mutant. It's absolutely essential for convergence and extension. So that was also a good candidate for regulating PCP pathway, but here is another killer experiment. Prickle still knows where the anterior is. Okay? So how does this gene, which is actually chromatin factor, and Margot demonstrated that thousands of genes are misregulated, yet some components of the patterning are occurring normally. Margot noticed that the notochord boundary in these mutants is affected and is uneven. And then we have done genetic experiments to test how these pathways, PCP and ODU, work together. And you can see that single mutants are short, and the double mutant is really, really, really short. This is consistent with ODU working in parallel to PCP pathway. And in this model, we can think about two influences. One influence being PCP, which polarizes all the cells. But what happens in ODU, the notochord boundary cells become mispolarized, and therefore maybe a cue from the boundary is also defective. And therefore, in the double mutant, the notochord cells become completely mispolarized, and other cells as well, leading to a very short axis. Margot demonstrated that part of the cue might be overexpression of certain molecules, such as EpCAM, and also integrin. So PCP pathway is polarizing cell behaviors, but it's doing this only on the dorsal side of the embryo. How does that happen? Again, going back to our mutants, we decided to look how BMP regulates convergence and extension. So we used very simple methods in which we uncaged cage fluorescein. This allowed us to actually measure how a simple cell population on the lateral side elongates and converges towards dorsal axis. And we could detect the cell population at the end of gastrulation and look at relative gene expression. You can see that in wild-type embryo, this very, very small cell population elongates very strongly and cuts through the somatic mesoderm. In BMP, in excess of BMP, like in Cordino mutants, cells are mispolarized, and you can see that both convergence is affected. Of course, cell fates are affected as well. There is much less of somatic mesoderm, but these cells, this cell population, instead of extending, is mostly moving towards vegetal pole. Opposite happens when we have too little BMP. in this dorsalized SMAD5 mutant, extension happens, convergence is also reduced, and somites form all around. What happens at the ventral side of the embryo? When we label this cell population, it moves to the vegetal pole, so there is no convergence or extension on the dorsal side of the embryo. And this we termed no convergence, no extension zone, which is increased in the ventralized mutants. By contrast, in the dorsalized mutants, on the ventral side, we have ectopic extension. With these and other experiments, we've demonstrated that BMP not only is a morphogen for cell fate specification, but is also a morphogen for cell movements, in such a way that at the highest level of BMP signaling, cells simply move towards the vegetal pole. In the lateral region, at the modest level of BMP signaling, cells are moving towards dorsal side and migrating, and at the lowest level of BMP signaling, they are undergoing intercalation. So I'll now tell you just a little bit about experiments that we are currently excited in the lab. And this is to, again, look at BMP with new tools that became available. And this is done by a very talented graduate student, Chateng Cha, and collaboration with Tony Tsai, who is faculty in the department. Chateng is using optogenetic methods, and we are using transgene to, at the same time, look at endoderm and mesoderm. And you can see that in the wild-type embryo, they move almost step in step. And in the ventralized situation activated with optogenetics, we can see that endoderm and mesoderm do the same. They mostly move towards the vegetal pole. So stay tuned to how endoderm knows where mesoderm goes to follow in convergence. So in summary, we've demonstrated that PCP pathway really is present all around the embryo to polarize cells and to provide a compass for cells to orient themselves with respect to the anterior-posterior axis. BMP is limiting PCP activity to the dorsal side in such a way that PCP becomes active only in the dorsolateral regions. And Vicky tells me that I should be finishing. So I will just mention to you that we are back to the screening, and we are finding some very exciting maternal effect mutants. And I'll tell you just and show you a movie about this very exciting mutant, which has not only is shorter, but also has a ear inappropriate location on the yolk. And Linwei Li, very talented postdoc, has named it Basset Hound. So how do ears end in the wrong location? You can see here that already during segmentation stages, the otic placodes are farther away from one another. And in this beautiful movie, you can see, and you will see otic placode with PAX8 appearing here in the wild-type embryo, and notice that somehow ear is sliding away from the dorsal axis. So stay tuned what this gene is in the future. So in the last moment, I will just thank people who are involved in these studies. This is my lab over the years. Been always fortunate to be joined by people who share my fascination with gastrulation, people who are collaborative and work together in the lab and beyond. And this is a list of some of the lab members who after leaving the lab, have started their own labs. I will just mention, I mentioned Margot, who is now at Baylor College of Medicine, Ryan Gray, who is here at the conference with beautiful presentations at Dell School of Medicine, and others as well. I will also mention my current lab members who are doing fantastic job both with gastrulation and disease models. And the last thank you is to my department, which also flew away very quickly. I wanted to say that we are all ambassadors of developmental biology, and we can all contribute in different ways, not only doing science, not only teaching, but also building departments and promoting careers of other developmental biologists. This is an amazing time in developmental biology, and if some people think we have a PR problem, I think it's a self-inflicted problem. Developmental biology is fascinating, it's very significant, and it is up to us to keep our head high and show it to other people. Thank you.